Mr. V, your video is muted. We can't hear you on the call.
Can you hear me now?
Yeah, we can hear you now.
Okay, great. Thank you so much. Well, normally I don't repeat the attendance words, but since you guys missed it for it being on mute, the first word for attendance is John Lennon. Happy birthday to the late John Lennon. Anyway, I didn't mention anything. You guys didn't mention too much. So I'm just reminding everybody that your egg or your sperm are called gametes. And then I kind of transition to this conversation that we're having right now, which is about the life cycle of multicellular organisms. In the context of this chapter, that's going to be from the adults of one generation to the adults of the next, right? So, my daughter is an adult, right? So once she has kids, that'll be kind of like the beginning of a new life cycle and the end of another in this context. Not that I'm going to test you on that, but it'll just make more sense when I show you the next slide and you know the context of the next slide. Another thing that's very important is this second bullet point right here. Having two sets of chromosomes, one from your mom, one from your dad, is a key factor in the life cycle. So here we go. Let's look at this. Here's some stuff you definitely need to know. Humans are what we call diploid dominant, which means that all of our body cells, which I mentioned earlier called our somatic cells, they contain pairs of homologous chromosomes. We already talked about what homologous chromosomes are. Remember, humans have 23 pairs of chromosomes, right? So we have 46 chromosomes. And the homologous chromosomes are pairs. So say, for example, chromosome number 7. For all humans, with some slight variations, chromosome number 7 is the same length, it's got the same centromere position, and it contains the same genes. Different versions of the same genes, but the same genes, generally speaking, right? That's a homologous chromosome. So humans, when we say diploid dominant, that means, again, most of our cells in our body are diploid, meaning they have both sets of chromosomes, right? Or as this reads here, your book also mentions diploid dominant and alteration of generations, but you do not need to know this. Know that humans are diploid dominant and know what diploid means. So again, diploid means, if you look at a cell that's diploid, that means it has a set of 2 chromosomes, right? Or its chromosomes come in pairs. So that is us, the majority of us, right? Now our cells, or excuse me, our gametes, our egg or our sperm, those are called haploid. You definitely need to know the difference between diploid and haploid. So haploid means you only have one of each of those chromosomes. So it's like half of the normal chromosome set. Now, there are some organisms out there that you can look up for independent work that are actually haploid dominant. Like, for example, actually, I guess I shouldn't give you an example, so you can look it up if you want for independent work. But yeah, some organisms, if you look at them, if you were to look at any of their cells, almost any of their cells, you would find that they only have one set of chromosomes. But then there are certain cells that actually have the whole set. So it's kind of like the opposite of us, because remember, most of our cells have the whole set of chromosomes, but then it's only our egg and our sperm that have half of the set, which makes sense, right? Because when our egg and an egg meets a sperm, we want to have that come together to have a full set of chromosomes. So half and a half equals a full. So in the human life cycle, what we have is a haploid sperm that fuses, with a haploid egg in a process called fertilization. At that point, we now have a new cell, right? When those 2 cells come together, and that new cell, that fertilized egg, is called a zygote. And again, since you have half and a half coming together to make that zygote, half and a half is a whole, therefore that zygote is diploid because it has the full set of chromosomes, one from each parent. So there's a lot of information on here. You definitely need to know this. Definitely know the difference between diploid and haploid. Definitely know that humans are diploid dominant, meaning almost all of our cells have the entire set of chromosomes. Definitely know that the exception to that is our gametes, our sperm and our egg, which have half of our chromosomes, right? Only one pair. And know when they come together in fertilization, it produces a diploid zygote. There would definitely be test questions about a lot of this information. Any questions about what's on this information? All right, the next word for attendance, pay attention, this one's gonna be a little bit trickier. Man, I can't talk today. It's gonna be sort of like gamete. The word is game, that's the inspiration, but instead of sending me the word game, send me whatever your favorite game is. Go fish, poker, football, Monopoly, whatever. World of Warcraft, I don't know, whatever your favorite game is, that's the next word for attendance, or phrase for attendance, if you will. Any questions? All right, I think the next slide is a visualization of this. This is from the old textbook, but it's perfect visualization. Right here, remember the life cycle as far as we're concerned in this context goes from the adult of one generation to the adult of the next. So here we have these two adults. All of their cells, because they're other than their sperm and egg, are diploid because humans are diploid dominant, which means we have 2n equals 46. Meaning that 2n equals 46 means we have 46 chromosomes, right? As you need to know. I've told you need to know that. So that's just repeating, reminding you need to know that information. And the 2n just means that when we have 46 chromosomes, that means there's two, it comes in pairs, right? So if you were to do some algebra and solve for n, right, you would divide both sides by 2, which means n equals 23. Meaning we have 23 pairs of chromosomes. We have 46 total chromosomes. Now, some of you probably didn't like me using that word algebra because you probably thought, oh crap, I hope we're not going to have to use algebra on this exam. And you're not. I'm just explaining what these words mean. I'm not going to use them in any of the exams. Not words, but the 2n equals 46. Just explaining what it means so you understand, but I'm not going to use that on any of the questions. Maybe in the quizzes, but not the exam. Anyway, so we have these diploid adults. Some of their cells go through meiosis to produce some sperm and some egg, right? And then later, those haploid sperm and eggs, which are n equals 23, right? Because they don't have 2 of each chromosome, they only have 1 of each chromosome, which is why it says n23. Those come together in a process called fertilization. The result is a diploid zygote. So again, we're back to the normal numbers, which is 2n equals 46, right? We have the full set of chromosomes. And of course, that diploid zygote goes through a lot of mitosis and eventually becomes a full-grown human, where the circle of life continues. Any questions about this slide? All right, so again, make sure for the exam, you know that humans are diploid dominant. Make sure you know what diploid means. Make sure you know that our gametes, well, first of all, know that egg and sperm cells are called gametes. Then also know that they're haploid. Make sure you know they come together in a process called fertilization. Make sure you know that results in a diploid zygote. All right, any questions about that? All right, so the next slide, we're finally going to start jumping into meiosis itself. So here we go. Chromosome duplication, right? That's the first part. Well, technically that's not meiosis yet, because as you guys know, just like in mitosis, the chromosomes duplicate during interphase. And let's see, is it up here? No, it's not up here yet. Actually, that's not even being shown. So in this picture, the chromosomes, no, they haven't duplicated yet. Okay. Interphase before meiosis, yeah. So yes, this is an interphase. Does anybody remember which part of interphase this is happening? So I'm glad it's not up there, because that will be a test question. It was also one of the pre-lab questions for this week's pre-lab. So when in interphase do the chromosomes duplicate? You definitely need to know this for the exam. And like I said, it was also a pre-lab question. So if you've done pre-lab, you should already know what this is.
S phase.
Yes, thank you, S phase. If you remember, you can add that to your extra credit words if you send those in today. Anyway, yes, it happens during S phase of interphase. And so far, that part's normal, right? Now we're looking at a pair of homologous chromosomes, right? One from your mom, one from your dad. We don't see all 46 chromosomes. We don't see all 23 pairs of chromosomes. We're just pretending like whatever organism this is only has 1 pair of chromosomes. And that's them, again, one from the mom, one from the dad. It goes through S phase of interphase. So the one from the mom duplicates, the one from the dad duplicates. And now we still have a pair of homologous chromosomes. But now for each one of those homologous chromosomes, we also have sister chromatids, just like we did in mitosis. So remember, in mitosis, that's the only thing we focused on was the sister chromatids. So we're still focusing on the sister chromatids, but now we also have homologous chromosomes. And that is a big difference. So for the exam, whenever there's any questions where you're comparing and contrasting mitosis versus meiosis, in mitosis, we're never going to talk about the homologous chromosomes. I mean, yeah, they're doing their things, but we're not worried about what they're doing in mitosis. The only time we ever talk about homologous chromosomes in the exam is when we're talking about meiosis. So let that be a hint for you for the exam. If there's any questions comparing and contrasting mitosis versus meiosis, when we're talking about homologous chromosomes, you should immediately think, that's meiosis. So anyway, S-phase, interphase, they duplicate. So that means they're about to go into meiosis, which is very similar to mitosis, as you'll see. Oh, this is like the Cliff Notes version. And also, I want to remind you, these pictures are not from your textbook. This is from the old textbook. So if you prefer these pictures over the ones in your textbook, you need to download this PowerPoint to see these pictures. But anyway, in meiosis 1, notice we skipped all the little middle steps, prophase, metaphase, anaphase, and telophase, because this is just an overview. So here we go. We have these homologous chromosomes. In meiosis 1, those homologous chromosomes separate. So now we have a cell that has only dad chromosomes in it and a cell that has only mom chromosomes in it. In this example, it doesn't always work that way. We'll cross that bridge when we get there. That's the first round of meiosis. Then we have another round of meiosis, meiosis 2, and then we separate the sister chromatids. So as far as we're concerned, with very few exceptions, and we'll talk about those exceptions, meiosis 2 is essentially like mitosis. Because remember, the whole purpose of mitosis is to separate those sister chromatids, right? So then you can have full-blown chromosomes. Same thing here. In meiosis 2, we're taking those those sister chromatids, and we're separating them. So definitely understand that. There's a lot of information on this one picture. Again, a lot of the questions on the exam for this portion will be comparing and contrasting meiosis and mitosis. So again, one of the things you need to recognize is when we're talking about homologous chromosomes, we're talking about meiosis. When we're talking about sister chromatids, especially when we're looking at them from here and here, sister chromatids, we could be talking about meiosis or mitosis. Also know that mitosis, there's only one round of mitosis. In meiosis, there's 2 rounds, right? 2 rounds of meiosis. That's another thing to compare and contrast. Meiosis, you start with 1 cell, you end with 2 cells. Excuse me, mitosis, I hope I said that right. Mitosis, you start with 1 cell, you end with 2 cells. In meiosis, like you can see here, you start with 1 cell, you end with 4 cells. In mitosis, you start with a certain amount of chromosomes, and you end with the same number of chromosomes. In meiosis, you start with a certain amount of chromosomes, like right here, there's 2, right? And then you end with half of that number. So there's a lot of stuff comparing and contrasting that you can see on this slide. And we haven't even gotten into the details yet. So are there any questions about this slide, this overview slide, before we get into the details of what exactly is happening in meiosis 1 and what exactly is happening in meiosis 2? All right, if there's no questions, I'll mention one more thing because it's not written on here. In meiosis, we start with diploid cells, we end with haploid cells. Unlike mitosis, remember in mitosis, we start with diploid cells and end with diploid cells. Any questions? All right, now that we've seen the overview, let's actually talk about the process of meiosis. So once again, in meiosis, we are producing gametes. Right? We're producing, the whole purpose of meiosis is to produce sperm or produce egg. Now your book is a little bit more specific and mentions that it's germ cells that start this process. It's not like you can just take a skin cell and turn it into a sperm cell, or take a kidney cell and turn it into an egg cell, right? It has to be germ cells. But when you do this, put an X to this because I don't care that you know that much detail. You can look it up for independent work. Like, what is a germ cell? Where are they located? I don't care that you know that, but you can look it up if you want. But what you do need to know is that meiosis produces haploid gametes. Another way of saying that is meiosis produces sperm or egg that have half the original number of chromosomes. So again, a lot of it's going to be comparing and contrasting. So let's look at these two major differences, right? Because meiosis is a lot like mitosis, but it does have these two major differences, and I mentioned them earlier in this slide, and now you can see it in writing. The first major difference is that the chromosome number is cut in half in meiosis, right? Because cells undergo two divisions, right? They go all through meiosis 1, where everything gets cut in half, and then they go through meiosis 2, where it gets cut in half again. So think about that. This is what the second bullet point is. You start with X amount of chromosomes, you double it, just like you would in mitosis. Then you cut it in half, which brings you back to the original number. Then you cut it in half again, which brings it to half the original number. Does that make sense? Just to use easy numbers. And humans, this is not for humans, because humans have a lot more chromosomes. If you had something with 2 chromosomes, organism that had 2 chromosomes, and it went through S phase of interphase, so it doubles, now it has 4 chromosomes. Then it goes through mitosis, or meiosis 1, right? So that number is cut in half back down to 2. And then it goes through another round, in this case of meiosis 2, and that number is cut in half. So that's how it works, right? Original number, double it during S phase, cut it in half in the first round of meiosis, cut it in half again. And that's just the easiest number I can give you as an example. For humans, obviously it would be a lot more than that, because we have 46 chromosomes or 23 pairs of chromosomes. So that's the first major difference between meiosis and mitosis. Anybody have any questions about that before we talk about the second major difference of meiosis and mitosis? Okay. So the second major difference, and this is a huge one, is called crossing over. And this one's gonna be a big one, because we're gonna talk about this in this chapter. We're gonna really talk about it in the next chapter. In this chapter, and the first time you get introduced to it, it's actually a pretty simple concept. And the next chapter is gonna be a little bit more complicated, and you'll see why. But crossing over, what is crossing over? It is the exchange of genetic material between homologous chromosomes. So what happens is, again, you've got a chromosome from your mom and a chromosome from your dad. And during this process of meiosis, they get really close to each other. And when they do that, they literally swap chunks. I don't know if you guys can see that way back there, especially since someone screwed the display settings, so you're not seeing the full screen, you're only seeing the small screen. But can you guys see that what's left here is like a mom chromosome with a little chunk of dad on there, and a dad chromosome with a little chunk of mom on there? Can you guys see that? That's what crossing over is. And that picture is just one way of showing it. So first of all, sometimes crossing over never happens. Sometimes it crosses over just like it shows right here, where, hey, there's just a little bit from one and a little bit from the other. Sometimes it happens more than once. So you might have a dad chromosome with 2 chunks of mom on it, and a mom chromosome with 2 chunks of dad on it. Sometimes it's just a little bit that's swapped over. Sometimes it's a lot. The point is, it's never just like that. It always varies. Meaning, at the end of meiosis, when you look at each one of your sperm and your eggs, right now, if you were to do that, if you could somehow look at each one of your sperm and egg and look at the genes or the chromosomes, all of them almost would be mostly one parent with a little bit of the other parent on there. And that'll be really important later when we talk about some other stuff. But for now, just know that crossing over just means they swap some genetic information. Meaning, at the end of this, for the most part, because like I said, sometimes this doesn't happen, but for the most part, at the end, none of those chromosomes in sperm or egg are completely from your mom and completely from your dad. It was a good time for me to say this too, because I just thought of this. I never said this in this context. When you go through meiosis, your chromosomes are split up. Your homologous chromosomes are split up, right? So when you look at any given egg or sperm, we're getting crossing over. If you look at each one of those chromosomes, some of them came from your mom, some of them came from your dad, right? So in a sense, you're not really giving them your chromosomes, because they're not your chromosomes. Half of them are your mom's, half of them are your dad's. So at the end, you're giving them either you're giving them a different variety, a different ratio of some of your mom's chromosomes versus some of your dad's chromosomes. And again, that'll be really important later. Later, I'm going to give you some math. And I know everyone hates math, but not math that you have to do, but math to explain the crazy chances of you being you. Like basically, if your parents, assuming they had sex and it wasn't IVF, but even then, if they didn't make you, exactly when and exactly how they made you, you wouldn't be you. Because the odds of that particular sperm, which is completely unique, and that particular egg, which is completely unique, meeting together and fertilizing is astronomically small. And again, we'll talk about that later. I find it fascinating, and once you hear it in context, hopefully you will too. Anyway, those are the two main differences between meiosis and mitosis. So are there any questions about the two main differences between meiosis and mitosis? All right, then let's move forward. Let's actually talk about the process of meiosis. There we go. These pictures from your old textbook again. Here's interphase. Everything's looking normal, right? You've got the nucleus in here with the full nuclear envelope intact. You've got the chromosomes all uncoiled, so you can't recognize one chromosome versus the other, right? Because they're just uncoiled. You can't tell. You got the centrosomes doing their thing. They haven't made a mitotic spindle yet. They're just kind of hanging out, right? That's interphase, right? In that process, what we don't see happening, but it's on here in words, is the chromosomes are duplicating, right? Because S phase is part of interphase, so that does happen. Here we go. This should look very familiar to you with just some other exceptions. Prophase 1, right? Prophase and meiosis 1. It's very similar to mitosis, right? So that's why I said earlier in the week, I would recommend learning mitosis before this, because when you know mitosis, you can just be like, oh yeah, this is similar, but there's just slight differences. But here's how it's similar. Once again, we have the mitotic spindle starting to form, right? Because those things have to, and what it's not showing is not only do the mitotic spindles form, but they also attach themselves to the chromosomes. Remember, that should make sense to you because the whole thing is all about moving chromosomes around. So the thing that moves them around has to attach to them. The nuclear envelope has disappeared, just like it does in mitosis. The chromosomes have coiled up, just like it has in mitosis, right? So all of the same things that happen in mitosis is happening here. The only difference, and I mentioned this earlier, is crossing over. And you can actually see it happening in this picture. So when we looked at the same picture of mitosis, we were just looking at random chromosomes, right, with their little sister chromatids, randomly. Here we can see that these homologous chromosomes are together, right? So here we've got, let's say this organism only has 2 pairs of chromosomes. There's pair 1, pair number 2, right? So pair number 1, you have the mom version and the dad version. Pair number 2, which happens to be a lot larger, you have the mom version and the dad version. They are together in this process, right? They're not necessarily attached. I don't even want to say that. Let's just think that they're together, right? Those mom and dad chromosomes, they love each other, so to speak, so they're staying together. And again, like we said in the last bullet point, and it's hard for you guys to see probably, but because they're kind of touching each other, they are crossing over. And I love this picture because, again, it's hard for you to see. In the last picture I showed you, they showed crossing over happening once. Here, in this particular picture, they're showing it happening twice. And again, like I said, the number of times varies. Sometimes it happens once, sometimes it happens twice, sometimes it never happens at all. That's a good independent work. And I don't know if I was ever taught this, but if I did, I forgot. Now I'm wondering, what is the average number of crossing over events for a human? Like if you were to look at a human chromosome during meiosis, on average, how many crossing over spots, events does it happen, right? So like this, like I said, this one was 2, the last one was 1. If you were to average those two, it would be 1.5. Hey, you can look that up for independent work if you want. Anyway, that's prophase 1. Now we move into metaphase. And this is, again, very similar to mitosis. In mitosis, the sister chromatids line up in the middle of the cell. And just to keep things different, for some reason, textbook authors almost always show mitosis up and down and meiosis horizontal. But it's all the same, right? A cell is a cell. It all depends on what angle you're looking at it from the microscope. But they do that for a reason, just to help your brain recognize that and say, oh yeah, horizontal, that's meiosis. But just so you know, in real life, that's all subjective. But anyway, here we go. In metaphase, in meiosis, it's the homologous chromosomes that have lined up in the middle of the cell. So now basically you could say they're lined up double file, right? I'll try to draw what I mean. If we were talking about mitosis, it would just be, like a single line of chromosomes lined up in the middle, right? Just a single line. And those X represents those are the 3 chromosomes right there. Each one of them has sister chromatids, which is why they look like an X. Single file line in mitosis. But instead, what we're looking at is meiosis. And here they've lined up double file, because the homologous chromosomes are still sticking together. They've already done the crossing over, by the way, and that's a test question. I should say that right now. When does crossing over occur? It occurs in prophase of meiosis 1. That will be a test question. Crossing over happens in prophase 1. Any questions about that?
Is there a limit to how much crossover can happen? I mean,
That's what I was just saying. I don't know.
don't know.
I either was not taught that, or I don't remember it. Or maybe scientists don't know. Oh, man, that would be even better if no one knows. Because then maybe I can do that research. Win the Nobel Prize. But no, I don't know. That's a great question. How many times can they cross over? Versus how many times does it happen on average? I would like to know that. Anyway, that's metaphase 1. Again, a big difference between metaphase 1 of meiosis and mitosis is that here we are dealing with homologous chromosomes. We're not even considering sister chromatids yet, right? Only homologous chromosomes. And like I said earlier, anything about homologous chromosomes on the exam, you should automatically think meiosis. Because when we talk about mitosis, we don't worry about homologous chromosomes. Since we're talking about crossing over so much, I'll throw another wrench in the plan here. The next word for attendance, name your favorite restaurant in Cross Lanes. And if for some reason you're not from here, don't be lazy on me now, be honest. If you're honest to God, don't know any restaurants for Cross Lanes, and just, I guess, put the word Cross Lanes. But surely you guys have been to Cross Lanes. And if you don't know, here are some fast food restaurants: McDonald's, Arby's, Burger King. Is there Burger King? Yeah. Taco Bell, Wendy's. Right. So if you have to, pick one of those. But yeah, so that's the next word for attendance. Is your favorite restaurant in Cross Lanes?
Is Golden Corral Cross Lanes?
I think so. Yeah, I forgot about that. Man, that is my son's favorite restaurant. We've been there like once.
Oh,
Anyway, so that's metaphase. Again, we're dealing with homologous chromosomes that have come together, not sister chromatids. Now, again, just like I mentioned in mitosis, right? They move to the center of the cell. What you can't see in this picture is that those mitotic spindles are pulling at those homologous chromosomes. It's like a tug of war. And then as soon as that tug of war rips them apart, then that is considered anaphase, because now the homologous chromosomes are separated. So now it is anaphase. Meanwhile, which you can't see in this picture too, it's not like everything is happening. This would be better in a video, right? So what's happening right now, if we were watching a video, they wouldn't just be apart, they would still be moving apart, right? They would still be moving towards opposite ends of the cell. And once they reach opposite ends of the cell, then we have the end, right? We have telophase. And of course, while telophase is happening, so does cytokinesis. So then we can see this cell is about to split into 2 cells. So far, that looks very much like mitosis, right? The only difference is, again, we're dealing with some crossing over of the homologous chromosomes in prophase 1. And again, we still have chromosomes lining up in the middle of the cell, but instead of being sister chromatids, we're looking at homologous chromosomes. But then again, they get pulled apart, they get all the way apart, and then that's telophase and cytokinesis. And again, at this point, now we have 2 haploid cells. And I know, again, it's really hard for you to see this, but you can download the PowerPoint. But if you look really closely, like this dad chromosome here has a little bit of a mom chunk on it. And this mom chunk has a mom chromosome has a dad chunk on it. So you can see like the crossing over has occurred, even though it's hard for you guys to see, because once again, someone screwed with the computer. Anyway, that's the first round of meiosis. Does anybody have any questions about the first round of meiosis before we talk about? No, before we look at all the things that we just talked about but in writing.
When telophase happens, but cytokinesis does not, is that when conjoined twins are produced?
Let me think here. You would have this first round of meiosis. Then you would have, let me think. That's a good question. So you would have one cell that has double the amount of chromosomes. I guess that could happen. Well, no, because then you would have, I'm sorry. If that were to happen, if we would have this mistake in this process, what that would lead to is some of your gametes having too many chromosomes and some of them having not enough, which is really bad. Usually it's deadly. Sometimes you get things like Down syndrome. So the process of twins actually comes from a different process. If it happens, if you have a mistake in meiosis, then that's leading to big mistakes where you have basically a zygote, like I said, that probably won't make it, or it'll be something like Down syndrome, where you have an extra pair of extra set of chromosome 21, or Turner syndrome, which is where you have like a female who has an extra X chromosome, things like that. But yes, that's what it would lead to, is extra chromosomes, not twins. Which is a great independent work topic then, if you want to look that up, because we don't even get into that to this class, because that process is more of like what happens during fertilization. So, great question. Plus, there's fraternal versus maternal twins. You can look up the difference between that. Any other questions? All right. Well, since she asked that question, let's just do another independent work. Another attendance word. You guys get a lot of points today, so I'm just throwing a lot out there. So for this one, name a set of twins that you know about, whether it's someone that you know or a famous set of twins. Like I can only think of like 2 celebrity twins off the top of my head. But yeah, so that's your next independent attendance word is a pair of twins that you know or know about. I guess they could even be fictional too. I can't even think of any fictional twins right now.
There were twins in Harry Potter.
Oh, see, I haven't read that. I was just a little bit too old for that, I think. But see, that something comes off wrong. I'm just old, is what I mean. So by the time those came out, I think I was in like, when did they come out? Mid to late 90s?
2000.
That late? Yeah, no wonder. So I was in the Marines. So even if I wanted to, it probably would have picked on, Back then, I actually cared what people thought. Anyway, prophase 1. Yes. So again, everything we just saw, but let's talk about it in writing. It's essentially the same as mitosis with some exceptions. Again, like we already said, with prophase 1, we're dealing with homologous chromosomes. We're not even concerned with the sister chromatids yet. Technically, when those things come together, that's called synapsis. I'm going to put it next to this because I'm not going to ask you that. But when you're reading the textbook and you read the word synapsis, I don't want you to think, what in the world? He never mentioned that. So yeah, when those homologous chromosomes are together, that's called synapsis. Again, like we said, crossing over occurs in prophase 1. Specifically, prophase 1. And once again, I want to remind you, I'm teaching from the old textbook with this because I'm making it easier. Your textbook technically breaks it down to prophase and prometaphase. But as far as I'm concerned, these things are the same thing because I'm just trying to make things easier for you. And again, it's different depending on what source you're looking at. I think for a 100 level class, we should just be prophase, metaphase, anaphase, telophase. I don't like these extras. But again, I teach from the textbook, so I want to make sure that if you're reading the textbook, as I'm sure you guys do all the time, I want to make sure you see that I'm recognizing your textbook says this. I haven't ignored it. We're just not using that terminology. So any questions about prophase 1 before we talk about the second round of meiosis? All right. Meiosis 2. This should look really familiar to you. So once again, here we have prophase 2. Nuclear envelope is gone. They're still gone. The chromosomes are still condensed. The mitotic spindle is formed, and that's what you can't see in this picture is that it's eventually going to be attached to those chromosomes, and then it's going to be starting moving them towards the middle of the cell. And that's what, once it gets to the middle of the cell, that we are now in metaphase 2. And just like in mitosis, this should look exactly the same. Here we have a single line of chromosomes, right? Because now we're splitting the sister chromatids. We're not splitting the homologous chromosomes. We are now separating those sister chromatids. So it's a single file line. We're pulling on them, Once they separate, that is then considered anaphase 2. What you can't see in this picture is that not only have they been separated, but they're still currently moving towards opposite ends of the cell. And then once they get to the opposite ends of the cell, that is telophase 2. And then, of course, just like in mitosis, you have cytokinesis, right? So those cells separate. The nuclear envelope reforms, which they don't show here. The mitotic spindle disintegrates, which they have shown. And then the chromosomes uncoil, which they haven't shown. But again, now that we're done with meiosis, you can see we have 1, 2, 3, 4 cells. It might be hard for you guys to see that, but there's this little line right here indicating that cytokinesis has basically happened. So they are separated. There is a division between them. They just haven't fully pulled apart. Yeah, so now we have 4 cells at the end of this instead of 2 cells like we did in mitosis. Okay. Any questions about that slide? Again, before the next slide, which is going to be some of the stuff I mentioned, but in writing. Nice and easy. All right. I need to erase this. That's wrong. We're not talking about metaphase 1 anymore. So now what we're talking about is, we're still talking about the second round of meiosis. I'm just going to M2. Meiosis 2, the second round of meiosis. It's essentially the same as mitosis, unlike the first round. The first round was a lot different. The second round is essentially the same. Sorry guys, this is in the wrong spot. We've already talked about this, but here it is again. So yes, metaphase 1, that is correct. Like we've already said, the homologous chromosomes meet in the middle of metaphase 1 as opposed to the sister chromatids. But then in metaphase 2, metaphase 2, it's the sister chromatids that meet in the middle. And here's another thing we need to know, and this will be very, very, very important later. When all these chromosomes meet in the middle in the first round of meiosis, all these homologous chromosomes, there's something called independent assortment. So, maybe I can visualize this for you. Good, we had different colors. Let's assume red is mom chromosomes and black is dad, or brown, whatever. Brown is dad chromosomes. I'll give you this quick example, then we'll move forward. And the reason I want to do this, and we're going to come back to it too, is in my experience, this seems to be a very confusing topic for people. Independent assortment means when those chromosomes line up in the middle, those homologous chromosomes, one from your mom and one from your dad, it's random. So here's what does not happen. Well, I guess technically it could happen, but very rare that this could happen. What did I say? Red is mom, I think I said. Let's say there's an organism that has 4 pairs of homologous chromosomes. When they meet in the middle, I guess technically you could have this. In metaphase 1, you could have it where all the mom chromosomes are lined up on one side and all the dad chromosomes happen lined up on the other. That could happen, but it would be very rare. What's more likely to happen is it just happens randomly. So maybe at the end of it, you actually have, there we go. I hope you can see the color difference there. That's the best I could do. See, what we have here is a mom chromosome, a dad chromosome, a dad chromosome, and a mom chromosome. What we have here is a dad chromosome, mom chromosome, and a dad chromosome. So think about all the different ways you have that combination, right? It could be all mom on one side and all dad on the other, or it could be mom, dad, dad, mom, or it could be mom, dad, or it could be dad, mom, right? However, think about all the different possibilities. Then think about the fact that humans have 23 pairs of chromosomes. If I were sadistic, I would make that an independent work topic for you until you finally realize that I was being really mean and it's impossible. Meaning I could say, draw all the possible combinations of our 23 pairs of chromosomes where it has moms on one side, dads on the other, and all the different ways it could happen. You couldn't do it. Like you wouldn't have enough time in this semester to do it. If you devoted the rest of your time to drawing those chromosomes, there's not enough time in this semester to draw all the possible combinations. So that is what independent assortment is. Again, when they line up in the middle of the cell, it's not like all mom on one side and dad on the other. They line up randomly. And again, This is going to be discussed later. It'll be a very, very important topic, and it'll also be a little bit more complicated than the way I just explained it. But that's a good introduction. All right, so again, we already saw this. Man, this PowerPoint needs some editing. Sorry, guys. Anaphase 1 and telophase. Yeah, these are all kinds of bad stuff. So again, stuff we've already said, stuff I've already said, but now here it is in writing. Again, I apologize for this being out of order. Mentally go back to the first round of meiosis. In anaphase 1 and in telophase, that's essentially the same as mitosis, except once again, if we're dealing with the first round of meiosis, we're talking about homologous chromosomes and not sister chromatids. All right, and again, this makes a little bit more sense since we're done talking about meiosis 2. This is everything I said, but again in writing. Meiosis 2 is essentially the same as mitosis, except the end products are cells with half the genetic material. Another way of saying that is meiosis produces haploid cells, right? And in mitosis, remember, mitosis, which is what we learned in the last chapter, you start with 1 cell, you end with 2. But in meiosis, you start with 1 cell, you end with 4. In mitosis, except for when things go wrong, we make a perfect copy of the cell. In meiosis, each new cell that is produced is genetically unique. And think about that. The reason it's genetically unique is because of independent assortment. Because it's not like, again, if you were to look at any 2 of your sperm cells or your egg cells, none of them have the same combination of mom and egg chromosomes. Because when all 23 pairs of them line up in the middle, they do it randomly. So the odds of you choosing randomly 2 of your sperm cells or 2 of your egg cells and looking at them and them having the same combination of like, all right, chromosome 1 was from your mom, chromosome 2 was from your mom, chromosome 3 was from your dad, whatever, so on and so forth, all the way to 23 chromosomes. The chances of that happening are astronomical. And actually, I'll give you the number of that later. And then that doesn't even include crossing over. So not only when you look at any given sperm or egg cell, will it have a different combination, but even if the small, tiny chance of them having the same combination of which chromosomes came from your mom or which came from your dad, then you have crossing over. So yeah, this sperm cell might have chromosome 1 from your dad, from your mom, and this sperm cell might have also chromosome number 1 from your dad. But because of crossing over, maybe this one has mostly dad with a little bit of mom, and maybe this one has about half dad and half mom, right? All because of crossing over. So once again, the numbers are astronomical, and we'll talk about them later. But yes, definitely know the difference between meiosis and mitosis. That is a lot of what this exam will be, this portion of the exam. Any questions about this slide? And if I remember correctly, this picture is not from your textbook. So if you want to study it, you need to download that PowerPoint. Same with this picture. Good grief. I don't know, I feel like somebody's messed with my PowerPoints. There's no way I'd make this many mistakes. So there's the same picture we've already seen. It's back. Anyway, with a minute left, we don't have time to jump into this next one. So when we get back on Friday, hopefully we can finish this chapter. Again, as a reminder, you don't have to come to lab today if you don't want to. I'll be here to help you. I'll be online to help you. The lab is due on Friday, as it usually is. And the last word for attendance is what you had for breakfast. If you had nothing, then the word is nothing. And that's it. See you guys at 10. Or not. It's up to you. And if you're online, don't forget to send those words before 9 a.m.